Hi, everybody. Um, we are doing chapter 7 in this video, and I'm finally on the fifth edition. And the question that the two authors posed for this one is, are you only as smart as your genes? So we're going to talk about uh, genes and heritability and passing on of genes.
So let's get into it. So we're going to talk about Mendelian genetics and quantitative genetics, which sounds really maybe intimidating, but it should be a pretty straightforward chapter. It's a short one at least, because next chapter we're going to look at genetics a little bit more. So we're going to talk about how do we get genetic diversity, difference between internal or identical and fraternal twins, dominant and recessive traits, genotype versus phenotype. We're going to look at Punnett squares and how things are inherited, at least in Mendelian genetics. We're going to compare that to quantitative genetics. And then single gene or polygenic inheritance, one versus many inheritance patterns, and then a little look at environmental effects, all to kind of answer the question about, is being smart something that you get from your parents? So the inheritance of traits, again, we're posing the question, are we basically copies of our parents? And we're not. There's certainly children are going to share similarities because they're getting their genes from the same gene pool, but each child is unique, and it's definitely not a copy of parents, and it's not an average of both parents. So if you remember, humans reproduce through sexual reproduction, which means we produce gametes by meiosis, which is what we talked about in chapter 6. Gametes, sperm, and the egg, they fuse to make a zygote, and then that continues to divide into an embryo, and that eventually becomes a fetus and a full-grown adult. Do you remember what genes code for? Do you remember what genes code for? Genes code for proteins. Genes code for proteins. And in fact, let me put, so you have DNA, and your DNA gets converted into messenger RNA. That way, the message of the DNA can leave the nucleus, and then it goes out into the cytoplasm where it meets something called a ribosome, and it gets translated into protein. DNA to RNA to protein. This is what we call the central dogma of molecular biology. So the DNA gets rewritten, it gets transcribed into RNA, because it's still nucleic acid, so it's still the same language, transcribed, rewritten. And then we go from RNA, which is made of nucleotides, into protein, which is made of amino acids. But this is the process. So if you have a gene on your DNA, it codes for a protein. Yeah. And I'm really not sure why they're talking about prokaryotes and eukaryotes and the shape of our DNA, but prokaryotes do only have one chromosome, and humans have 46. Oh, we are diploid organisms, so let's practice. 46, our gametes are haploid, which means they contain how many? 23. So, this is a nice little analogy that they built, where our genes are the instruction manual, and so you could consider this like all your DNA, and the chromosomes are individual pages, and each page is going to have thousands of words, and those words can be read differently to create different proteins. And then, of course, when you're making different alleles, so the way we can end up with some genetic diversity can happen in a few different ways. It can happen with mistakes, so mutations in the DNA. Remember, I said DNA mutations aren't always bad. This could just mean that you go from gray with an E to gray with an A, or strong to string, or you could end up with a mutation that doesn't make sense, and you end up with a word that doesn't make sense. You can also get genetic diversity during homologous pairing. So remember in oh gosh, I want to say it's well, it's in meiosis. I want to say it's in metaphase, where the chromosomes are lining up, and condensing and replicating so that they can separate into the first separation into individual cells. Um, what can happen there is, as they're lining up, similar genes, homologous pairs, can swap places on the sister chromatid. So that's one way. And then there's another way. When the chromosomes line up, there's not like an order that they line up with, like always this one goes on this side and this one goes on this side. It's called random assortment, so independent assortment during that process. And so it kind of is like reshuffling all the pages. As long as you get one copy of every page, it doesn't really matter which one it came from. Oh yeah, so I copied this slide over from this chapter 6 lecture to point it out. So crossing over happens in prophase 1 of meiosis 1. So that's when the little homologous pairs cross over, and it's so tiny and hard to see down here, but notice the little tips are not all red. This one's got a blue tip and this blue one's got a red tip. And then eventually during meiosis 2, you end up with them separate. And then also random alignment. I said, like, it's not always one on one side and the other on the other side. So like here, the two red ones are on the top and the two blue ones are on the bottom, but truly randomly, it could be red, blue, blue, red. That's what that means. Oh yeah, and here's a nice full picture of, so this is where they're swapping, where crossing over can occur, and then random alignment. So those two things can contribute to diversity in addition to mutations. So when the parents' genes are known, we will always know the genes of a child. Remember, I cautioned you, if you ever see the word always in a true false question, yeah, false. I mean, unless it's like a known scientific fact, like, what goes up will always come down, right? Well, even that won't work unless you're in space, right? Anyway, all right, focus. So we have a lot of genetic diversity. There's about 8 million different combinations, and then once you end up with random fertilization, so remember those two gametes have to meet, you end up with trillions of different possibilities. So I hate the way they post these questions, because some of them are review and some of them are like before we discuss the thought. But remember, we're supposed to know the difference between identical and fraternal twins. And so when we're talking about twins here, mono, do you remember what that means? It means one, right? So monozygotic twins occur when two separate eggs fuse with different sperm. No. So monozygotic twins are going to happen when one egg and one sperm join, and you have one zygote, a monozygote. But then instead of that just continuing to divide into a full fetus, it splits at the embryo stage and you end up with two embryos. That's how you end up with identical twins, because it was the identical genetic copy. Whereas fraternal twins are two eggs, two sperm, two completely random possibilities, and they just happen to be fertilized at the same time and carried at the same time. So Mendelian genetics. This is Gregor Mendel. He was a friar, and he was a meteorologist and just kind of an all around curious person. And he is kind of considered the father of genetics because he started to study crossing in pea plants, and he started to discover some patterns which he documented, and so it kind of became the foundation of genetics as we know it, although there are some nuances that we're going to learn. So when we start with Mendelian genetics, we have these very like left or right cut and dry observations. And so you have the genotype, and the genotype is the genetic composition. So it's the combination of alleles that the parent cell has. And the phenotype is what you see. So, for example, humans, we are diploid, meaning we have two copies, and yet we only see one trait. And so when you're talking about the genotype and you're talking about those two copies can be the same. So they would be homozygous, homo meaning same, or they could be heterozygous, meaning one copy of one allele and another copy of another allele. Um, and and so, for example, the alleles that Mendel noticed were possibilities were a wrinkly seed or a round seed, a green seed or a yellow, a white flower or a purple flower, a tall plant or a short plant. And when you would cross those plants that had those phenotypes, this is the observable trait, um, you would notice that there would be a dominant phenotype. So that means that the phenotype is seen when obviously when they're the same. When you have the same alleles, you're going to see that allele, you're going to see that phenotype. But when you have two different phenotypes, the dominant one is the one that produces the phenotype. That's the one that you see. Whereas the recessive one is the one that's like hidden, that yes, the person has that allele, but that's not the one that we see. And a Punnett square is a way for us to kind of draw these type of crosses. So when we're talking about, you know, alleles, we give them a letter to kind of denote the trait. So in this case, cystic fibrosis, F for fibrosis, and the disease is a recessive disease. So and F would be like no fibrosis. And so if a person it is homozygous, they would have capital F. And if a person was recessive, they would have lowercase. And in this case, here with a capital and a lowercase, oops, I didn't mean to click. That person is heterozygous. They have two different alleles. And because the dominant one, which we put as capital, is for not having cystic fibrosis, this person would not show cystic fibrosis. And what they're asking here then is, okay, if you had two parents, both that did not have cystic fibrosis, but because they each had the dominant allele that said no fibrosis, but they also each had an allele that was recessive that could potentially end up in a zygote in the offspring, that means that they would have cystic fibrosis. What options are there? And so we draw this square, this Punnett square, and we imagine that you separate those alleles, right? Because they end up with one allele in each gamete. And so you have a capital, a lowercase, a dominant, and a recessive, and then you cross them. So if you have a dominant dominant, the phenotype is going to be no fibrosis. And then you're going to cross this one with this sperm, and you would have dominant recessive, and again, no fibrosis. And same thing here, dominant recessive, no fibrosis, because it has the dominant trait is no fibrosis. But if you happen to get a recessive allele with a recessive allele, then you're going to have cystic fibrosis. And so the way this can be predicted then, if you have the Mendelian genetics of a dominant recessive alleles in this way, you draw a little Punnett square, and you can count, you categorize each of the alleles, and then you count the phenotypes. So in this case, 75% of these particular offspring do not have cystic fibrosis, 25% do. Now we have to add in one more thing. We have to add in something called a carrier. A carrier are the people that have the allele but don't show the symptoms. So just like in the case of cystic fibrosis and Huntington's disease, the alleles that have the dominant recessive here are carriers, just like the parents actually in this case, because the parents were heterozygous with the recessive allele. So here in the Punnett square where they proposed Huntington's disease, they had a carrier father with a dominant recessive traits, and the mother was, oh, I'm sorry, this one's backwards. This one, it is a dominant trait. So Huntington's disease is dominant. And so if the father has Huntington's disease and is heterozygous, and so if he's crossed with a mother who's unaffected, 50% of the children will be unaffected, but 50% of them will. And then 50% of them will also be affected. Yeah, here's a bit about Huntington's disease. I think maybe I put those slides in opposite order. Okay, story problem time. A pea plant has one recessive allele for wrinkly seeds and one dominant allele for smooth seeds. So we use the same letters. So dominant for smooth, and then the recessive would be a small s, and we would know that would be wrinkly. A plant has one recessive allele for wrinkly seeds and one dominant allele for smooth seeds. What would the plant look like? So what's the phenotype of this plant? If the dominant allele is smooth, yeah, it's going to be smooth. So that's Mendelian genetics, where you have, half and half. But quantitative genetics is where you have continuous variation. So you have a much larger range of phenotypes. It's not like you're either 5'3" or 6'5". That's the end of it. But you have this range in between. And those variations are a little bit more tricky. They're due to both genetics and environmental differences. And so, you have an average, and then you have the variance between that average, which is the range. And then you can have things that are polygenic. So more than one gene influences the phenotype. So eye color is one. It's not just like Well, because you know there are multiple colors for eyes, right? You have blue, brown, green, hazel. I'm sure I'm gray. You know, I'm sure you can count them in many different ways, because we have many different phenotypes. But there are, you know, a finite number of genes, but they count towards one observable trait. And then environmental effects. So yeah, genes do stuff, but then your environment does stuff too. So these are identical twins, so they were monozygotic. And the twin on the right was a lifelong smoker and went to tanning beds and things like that, and the other one didn't. And so you can see like the expression of the genes, they look similar, but then like, you know, the environmental effects of this one seems to be a little bit more wrinkly, um, different skin color, things like that. um, and most of our traits are going to have that continuous variation. It's really hard to separate genes from environment. But here's one. Can you pick the one that is for sure um, a uh, quantitative trait that isn't going to show continuous variation? Well, this kind of gives it away, right? Presence or absence. So like that's not a continuous variation. That's this or that. Now, heritability. So we're still trying to answer the question, you know, are you smart because your parents are smart? Heritability is, you know, it's what's available in the gene pool. So how likely is it for a particular genetic trait to be passed on to individuals? And there are some correlations that are used by human groups um, trying to sort out IQ to see if it's heritable. But again, correlation doesn't equal causation. That's really hard to say that, you know, you're smart because your parents are smart. Certainly, there are some things that are genetically passed on, and some things that aren't. And so one of the fun studies is looking at like twins and seeing if, and they call this like the nature versus nurture. So like nature is, you know, is that your genes and did those genes give you this IQ, versus nurture, meaning like, did you grow up in an environment that nurtured having a high IQ and gave you the opportunities to expand that and demonstrate that. And again, IQ, is IQ even a measurable you know, form of smartness? We use it, but that might not be an absolute. Smartness might be something hard to measure too. So in general, the answer is, you know, it's probably not likely that IQ is really all that heritable, although some things are definitely heritable. So it doesn't tell us about, you know, individual differences between between siblings. It doesn't tell you about individual circumstances. And just because a trait is highly heritable doesn't mean that it's automatic, that means that you're going to get it. Oh, okay. So, yep, this is the summary. Genes have a strong influence even on complex traits, but independent assortment and that crossover is going to give you a lot of different phenotypes. Environment has a large effect. And then you have these quantitative traits, which is really hard to predict. And so hopefully you are doing okay with those learning outcomes, and you can go back through them and you can speak to each of these. All right, well, that is the end of chapter 7. Chapter 7 is the introduction to genetics, and then chapter 8, we're going to talk much more about